So in this video, we're going to be talking about the types of inheritance. So we're going to be going over Punnett squares, pedigrees, and kind of how they help us to determine what our offspring are going to look like or how traits are going to be passed down over time. So the types of inheritance we're going to talk about, about both Mendelian and non-Mendelian inheritance. Mendelian, obviously the ones that Mendel worked with, monohybrid crosses, and dihybrid crosses actually should be up there. But our non-Mendelian traits then are going to be a little more complex, things that Mendel didn't really think of whenever he did his work with peas. So things like codominance, incomplete dominance, sex-linked inheritance, and multigenic traits. So the first one, monohybrid crosses, is just going to be the inheritance of a single trait. So we're just going to work with one trait to start out with. So if we have a plant that produces yellow peas, a plant that produces green peas, we can say that the yellow pea is homozygous dominant. So this means that there are two dominant alleles in this pea. So there are two big Ys. In our green one, we get a homozygous recessive. So we have two little Ys. Those little Ys are going to code for the other form of the trait. So this one is also going to be repressed if we were to introduce two big Y, or a big Y. So we are following the trait, or following this trait to see how it moves to its offspring. So if we combine this on a Punnett square, we have our one parent on the top and one parent on the side. We're going to create this four square where we drop two of these traits down and we move two of them across. So you can see in each box we get a big Y, a dominant Y from one side, and a green Y or a recessive Y from the other. So all of the offspring in this case are heterozygous. Heterozygous means that it has one of each trait, a big Y and a little y. So if we were to do an analysis on this and figure out, okay, what's the chance of us getting a yellow offspring? Our genotypes are going to reflect that because there is a dominant Y in each of the trait, or each of the offspring, there is a 100% chance that we are going to get a yellow offspring. We wouldn't get a green offspring because even though they have a recessive trait, that dominant trait is suppressing them. It's pushing them out of the way so that it gets all of the expression and all of the attention. So this is what we call complete dominance. Complete dominance just means that the one trait is going to completely overpower the other. So if we get a case where it is a heterozygous or a homozygous dominant, you are always going to see the dominant trait appear. So let's go over an example. So these are the mantises that I have in class. These are called ghost mantises. And in ghost mantises, we have a rare recessive trait that is that causes the head crest, this little part on their heads, to be really skinny. And this is called an illudens morph. So this was once thought to be a different species. We had Phylocrania paradoxa was our normal ghost mantis. Phylocrania illudens was our other form. And scientists figure out that, okay, these are the same organism, even though they have different shaped heads. So this is what we're going to call an illudens morph. And if we have two heterozygous individuals for this trait, how many offspring will have the illudens morph, or what percentage of the offspring will have the illudens morph? So if we have two heterozygous individuals, heterozygous means that we have one of each trait. We have a dominant and a recessive. So I'm going to use G because we have ghost mantises, and it's really easy to tell big G from little g. So our dominant trait is going to be this normal head crest. If we have a little g, that's when we're going to get this illudens morph. So we're going to get this skinny head crest. So when we take our parents, both individuals are heterozygous, meaning that they have one big G and one little g. And when they cross, the big G will drop down, the little g will drop down, the big G will move across, and the little g will move across. And in total, we will get one of the four of these four potential outcomes. We will have 1 in 4 chances that we will get a Illudens ghost mantis. The other 3, even though we have 2 that are heterozygous and 1 that is dominant, each of these 3 are going to produce a normal head crest because they all have that dominant trait. So this is what Mendel figured out with his P's. Same thing. His P's were really easy to follow traits because a lot of them were in this Mendelian format where there was just one is always dominant to the other. So, take a minute, jot down this problem and see if you can figure it out. Suppose we made a black wool sheep with a white wool sheep. If black is the dominant allele, what will the possible offspring look like? So take a minute, pause the video, solve the problem, and then we will go over it. Okay, so there were actually 2 potential options for this one. Because we say that black is dominant, so we're going to use big B, little b. So big B is black, little b is white. You notice that even though we have black and white, both of them share the same letter because they are the same trait. We're just worrying about wool color right now. If we were doing something else, we could use different letters, but right now we are focused on just one letter. So we have a black wool sheep and a white wool sheep. We know that the white wool sheep, if it has to be white, it has to be double recessive. So our white wool sheep is going to have this genotype, little b, little b. Black wool sheep, however, there's 2 potential options that you could have come up with. So as long as, since this is complete dominance, as long as it has a dominant trait, it will be black. So we could have either big B, or we could have big B, little b. We could have a dominant homozygous, or we could have a heterozygous with the dominant trait. So your Punnett squares should look something like this. If we cross a white sheep, regardless, it's going to carry that little b, little b. Both options both have this dominant trait, however. So if we cross them, if we cross a homozygous dominant, we are always going to get a black sheep, 100% of the time. If our offspring is or if our black sheep is heterozygous, though, we have a 50% chance of getting white because we have that black trait that gets passed down half the time and a white trait that gets passed down half the time. So there's a 50-50 chance that you can get a big B, or a big B, little b. Or big B, little b, or little b, little b. Okay. So as I mentioned, these types of problems that Mendel figured out how to do are really good at figuring out the probability of genetic crosses. Probability just means the likelihood that a particular event is going to occur. So for example, if I take a penny, I flip it in the air. The chance of it getting either heads up or heads down, so either heads up or tails up, is 50%. There's always a 50% chance that half of the time you're going to get heads up, half of the time you're going to get head down. But what if we combine 3 of them? Well, we take the 1/2 from the first flip, second half is going to be independent of the first flip, so it's going to be a half, And then the third one is also going to be independent, so 1/2 times 1/2 times 1/2 is 1/8, or about 13% of the time. So we can use this in our genetics to figure out what the chance of us getting a certain trait is going to be. For example, in this one, we start out with 2 tall plants, but both of them, it says over here, are heterozygous. So if we cross them, we have a 1 in 4 chance of getting a homozygous dominant, 1/2 chance of getting heterozygous, and a 1/4 chance of getting homozygous recessive. When we translate that to our phenotypes, our physical characteristics, we have 3 chances of getting tall and 1 of getting short. And this this improves as you add more offspring. So typically the larger number of offspring that you get, the closer your results are going to be to the predicted value. So if we just cross 4 plants, there may not be a exact 3-to-1 ratio of getting 3 talls to 1 short. If we breed, let's say, 300 plants or 3,000 plants, then they will start to meet that 3-to-1 ratio. It'll be a lot closer. So this is really simple when we're following 1 trait, but what happens if we get 2? This is called a dihybrid cross. This is where we're going to follow 2 different traits. So the way we do this, kind of like if you have seen in math class, we are going to use the distributive property to figure out what the potential offspring or the potential gametes are going to be for each offspring. So the way we do this, so we start with the parents. We need to figure out what the genotypes are for both parents. So if we cross one that has, let's say, heterozygous for size and pod color, we need to figure out that we have 2 traits, one from mom, one from dad, for each different gene. So the gene for pod color in this case is going to be different than the gene for the size. From there, we need to figure out what the gametes are going to be. So this is where the distributive property comes into play, because according to our laws of segregation and our laws of independent assortment, the chromosomes are going to separate so that there is one randomly chosen chromosome or one randomly chosen allele in each gamete. So of our 4 gametes that we could produce, there are 4 different options that could be there. So if we have a big T, little t, and a big G, little g, the big T and the big T and the big G go together, big T, little g go together, little t, big G go together, and little t, little g go together. So we have 4 different options that we could produce. From there, we line them up, same thing we did with our normal Punnett squares. You write down the new genotypes, and from there you can figure out the results. So again, if we're doing complete dominance, you can figure out that as long as it has a big capital letter, it is going to have that dominant allele and therefore that dominant trait. So if Mr. Howell wants to create a flower for his plant class that he wants to use for corsages. Corsages are just the little flower wristlets that you use for things like prom or dances. He wants to grow blue flowers for the school colors that are short and that are easy to work with. So he crosses a pure, short, white flower with a heterozygous, tall, blue flower. What is the probability that Mr. Howell would get a short blue flower? So blue in this case is dominant to white, and tall is dominant to short. So let's start with the parents. We say that B is going to be our color, and T is going to be our height. So blue is dominant. That means blue is big B. White then has to be little b. Big T, if tall is dominant to short, big T is going to be tall, and little t will be short. So our parents, if we have a pure, short, white flower, pure is going to indicate that it is homozygous. So we have a short, white flower. Short is our recessive, so it's going to be little t, and our color is white, so we want to get two recessive traits for that, so little t, little b. Our other flower is tall and blue, so it says it's heterozygous. We're going to consider that to be heterozygous for both traits. So heterozygous tall has to have one of each, and heterozygous blue has to have one of each. So these are our parents. And from that, we can figure out what the offspring are. So if we write down over here, we had our little t, little b, and our heterozygotes. We need to do the distributive property so that we can get one of each trait in each box. So the way that I've been kind of teaching it, just so you can keep track, is you can label them as 1, 2, 1, 2. Same thing with this, 1 and 2 for each type of trait. Because remember, you need one set of traits from mom, one set of traits from dad, so each of these alleles came from a different parent, and we're going to do that to figure out what our offspring are going to look like. So we're going to put the recessive on the top. So we're going to start with T1 and B1. From there, we need to get T1 and get its other cross out of the way, which would be B2. So again, using that distributive property, we take T with 1 and 2, and then we take T2 with H1. So while this doesn't actually do anything for the Punnett square itself, this just helps to kind of keep track of which crosses you've used and which crosses you haven't used. So then looking at our side column, T1 is a dominant T, B1 is a dominant B. Let's do this. So these are T2s, T1, B2, T2, B1, and T2, B2. So now comes the bit repetitive part of crossing all of these. So just like we did with the 2x2 Punnett squares, the 4x4 Punnett squares are going to do the same thing. So we take each trait and we cross it. So we're going to take the big T's across, take the little t's across, take the big B, little b. So let's actually take this down first. So take the t's down. 100% of the time for this one, we're going to get recessive t's coming down. And then we've got big B, little b. Big B, little b. So overall, like I said, a lot of writing, but from what we've done, we can figure out now what the chance of Mr. Howell getting a short blue flower is going to be. So we figured out that blue, if it has a big B, it's going to be dominant, which is how we get our blue. So this one's already out, and this one's already out because they do not have a dominant B. So we are left with row 1 and row 3. If we look at our T's, we want a recessive pair of T's. So we have a 1 in 4 chance of getting that. Now, some of these may not come out this cleanly. You may have one chance down here in the bottom right that it's going to produce a short blue flower, or somewhere in the middle. But in this case, we got lucky, and our middle column, or middle row there, is going to be blue and short. So, try out another example. We have a female guinea pig with short black fur and a male guinea pig with short white fur. The female is heterozygous for both traits. The male is homozygous for short hair. If black is dominant over white and short is dominant over long, what are our offspring going to look like? So take a minute, jot this down, and try and solve the Punnett square using the trick that we used in the last example. Okay, so our female and male genotypes, first thing we need to figure out. If we have a female that is heterozygous for both traits, it's going to get a big S and a little s and a big B and a little b. Our male, on the other hand, is homozygous for both traits because our white fur is recessive, and our short hair is dominant as well as homozygous. So moving this over, using our distributive method, our possible gametes for the female are going to be S1B1, S1B2, S2B1, and S2B2. Our male gametes are all going to be big S, little b, because we only have 2 big S's and 2 little b's. So we move that over to our table, and we cross them all. We get a bunch of them. So in terms of getting short hair, since it is dominant, all of the ones with, in fact, all of these will actually get the short hair because they all have a big S. They all have a dominant trait. In terms of getting the hair color, we have a chance over here to get white fur color, and a chance over here to get white fur color. So we have a 50-50 chance whether we get white or if we get black fur color. Okay, so what happens if one of those alleles is not completely dominant over the other, or if we have several alleles? So this is more of those non-Mendelian traits, because he didn't think of that. A lot of P traits are very simple and very easy to predict using basic Punnett squares and normal complete dominance. But then we get into things like incomplete dominance. Incomplete dominance is where we have a trait that is neither dominant nor recessive. So instead what's going to happen is the heterozygous genotype is going to lie between the two homozygous genotypes. So we have a red parent and a white parent. Normally, with complete dominance, we would have the red one overpower the white one, and all of these offspring will be red. But in this case, it's not what's happening. We have 4 chances of getting a pink offspring. So we cross the red and the white to get pink. So if we wanted to create a flower species that develops pink flowers, we start with a pure red and a pure white. We have incomplete dominance. We cross a pure red with a pure white. What's the probability that we get pink? So let's assign R as our color trait. If we have 2 big R's, we're going to be red. If we have to little r's, we will be white. And we want to be on the lookout for the heterozygous individual, the one that develops pink flowers. So we need to be on the lookout for big R, little r. So we will fill out our table. A pure red, we said, is going to have 2 dominant traits. A pure white, since it's homozygous, is going to produce 2 little r's. So if we cross them, same thing we have been doing, we will find that there is a 100% chance that we will receive a pink offspring. There is a 0% chance that we will get a white or a red, because none of these are going to have 2 dominant alleles or 2 recessive alleles. The next one we're going to talk about is codominance. So codominance works very similarly to incomplete dominance, but in codominance, co would be together. So if you collaborate, you work together. If you are in a co-ed school, you are working with both males and females. So co is going to be together. So codominance, rather than having either one overpower the other or that mix that we saw in incomplete dominance, our phenotypes are going to mix with this one. So if we have a white chicken and a black chicken, let's say that white is, we'll call this big W, black is big B. A speckled chicken is going to have 1 of each trait. So we can see this in blood typing. So if you have ever donated blood, you have likely figured out your blood type, or they told you your blood type. But blood types are codominant. So we have our recessive trait, little I. If we have 2 of those, we are going to be considered an O blood type. If we have one of our dominant traits, which we have 2, we have an IA and an IB. If we have an IA, then we are going to be an A blood type. So this could be the homozygous dominant or the heterozygous. B, same thing, but we are using the B allele, not the A allele. So big B, or big B, little I. If we are AB, though, we have 1 of each dominant trait, so both of them are going to be equally expressed as an AB blood type. So going through an example, what is the chance that you will have type AB blood if your mom is heterozygous for A and your dad is heterozygous for B? So if we break it down, we have our dominant I and our recessive I. We have IA and IB. So if we are heterozygous for A and heterozygous for B, that means we have 1 dominant trait and 1 recessive trait, which means that mom has to be IA and little I. Dad has to be IB and little I. So if we cross those 2, you'll see that we get 2 dominants in the first box, dominant and recessive in the second one, dominant and recessive in the third one, and 2 recessives in the fourth one. So if we want an AB blood type, we have a 25% chance of receiving that trait. Only 1/4 of our boxes give us a trait that is codominant for both A and B. So this also works with, like I said, color patterns or things like that. There's a couple different traits that use this, but this one is going to work so that both the A and the B are expressed. So the next one we're going to talk about is having multiple alleles. And what we aren't going to do Punnett squares for this one, it's good to at least go over. So multiple alleles means that many genes are going to exist in a way that there are many different alleles for the same trait. So for example, if we wanted to determine our hair color, you have 11 different alleles that code for your hair color. Rabbit coat color has at least 4, but you can see down here we have a lot of different colors and variations that can lead to different hair colors. So we aren't going to look too much into this in terms of Punnett squares because having 11 alleles is very difficult to map out on a Punnett square, as well as polygenic traits. So polygenic traits are the exact opposite. We have many traits, or we have traits that are going to be produced by multiple different genes interacting. So instead of it being one gene with multiple alleles, we have multiple genes coding for the same trait. So these traits are going to be controlled by 2 or more genes, thus poly is many. So for example, your skin color is based on a lot of different genes coming together. For example, melanin is going to determine, or melanin production is going to determine how dark your skin is. Different pigments are coded for by different genes. So all of these are going to determine how light-skinned or how dark-skinned you are. The last one we're going to talk about is sex-linked traits. So now, talking about this like we did in meiosis, there are traits that are carried on your sex chromosomes. So all of your 44 of them are called autosomal chromosomes, meaning that they are used for different bodily functions, different things like that. Sex chromosomes, X and Y, are going to determine your gender and a couple different things that are produced as a result of your gender. So females typically have 2 X chromosomes. Males have an X and a Y chromosome. And when we talk about sex-linked traits, most of the time the trait is going to be carried on the X chromosome. Now, why is that? Well, if we take a look, so this is the karyotype of a female. Karyotypes are just maps of our chromosomes. So you can see chromosome 1, chromosome 2, chromosome 3, all the way down to chromosome 22, and then we have our sex chromosomes at the very end. You can see in this female there are 2 X chromosomes. Both are about the same size, same centromere location, so they are homologous. But they have enough room that they can fit some traits on them. If we look at a male, however, males, again, are going to have all of those homologous chromosomes, but we have an X and a Y chromosome. And while there are traits on both of these, more traits are going to be held on the X chromosome than on the Y chromosome, which means that most of our traits that we're going to look at are going to be involving the X chromosome, not the Y. There are Y-linked traits, but those are very rare because they only pop up in male individuals, and they can only pop up because we have 1 Y chromosome. So sex-linked traits, when we look at them, we are going to do it kind of like the blood types, where we have an X and a Y with a superscript letter. So you can see on this sperm, we have a big X, or we have an X chromosome with a big H, a dominant H. We have a Y chromosome because all males carry Y chromosomes. Our female is going to have 2 X chromosomes. One has a dominant trait, the other one has a recessive trait. So you can see the difference between these two based on what case the superscript is. Kind of like if you're writing a like something to the 2nd power to the 3rd power, that is called a superscript. So whenever we cross them, we are going to take both the X and the trait and move it across, or the Y and the trait, well, the Y and move it down. Y's do not get the trait. So you can see we have a 50% chance of getting a girl and 50% chance of getting a boy. And whether or not something is going to be affected is whether or not it has, in this case, this recessive trait. So you can see that in this one we have a heterozygous individual, so they have the trait, but they are not going to be affected. In the boys, however, one of them has a recessive allele and a Y chromosome. If you have a Y chromosome, that means that as long as you have one trait, you are going to get it. So, for example, this one says hemophilia. So this individual in the bottom right is going to be affected by hemophilia because they have that trait. Even though she does not, he will. So this is why X-linked traits are more likely to appear in males, because females need 2 of these traits in order to have the to be affected. Men only need 1. So it's more likely that a man is going to be affected by a trait than a female. So let's put this into context of genetic diseases. So a mother is a carrier for male pattern baldness. Male pattern baldness is a trait that usually runs as a recessive sex-linked trait. What's going to happen is this is where you're going to progressively lose your hair. So male pattern baldness is going to be carried by this female. That means she is heterozygous. And the male, if he has the trait, that means he has to be X. Let's say baldness is big B, or normal is big B. So then our X little b has the baldness trait. So male pattern baldness is an X-linked recessive trait, meaning we have to have 1 or 2 of these X chromosomes with the recessive trait if we're going to get it. So if we have 4 children, what is the chance that they are going to have male pattern baldness? Her mom, we said, is heterozygous. She is a carrier, so she has a big B and she has a little b. The male has an X and a Y chromosome. We said the Y does not get anything in terms of a trait, but the X has to be little b because he is a he has the trait. So if we cross them, we are going to cross X's. I like to do X's and Y's first, but that is just personal preference. And then we are going to figure out the traits. So the big B will come down, little b will come down, little b will come across, and then the Y chromosome doesn't, so it doesn't transmit anything across. So in total, of these 4 children, we have 2 individuals that are going to be affected by male pattern baldness. We have a female that has both recessive traits. If we have both recessive traits, that means we are going to have the trait. And we have a male that has 1 trait and a Y chromosome, which means that he is also going to be affected by this trait. So we have a 50-50 chance of whether or not we are going to get an offspring with this trait. So now that we are able to better predict these types of problems, please go through on Schoology. There is a Punnett square practice as well as a duck genetics video, or ducks genetics activity. I would like you to go through those, try out what we just learned. Please use your notes and resources to figure out the probabilities for the Punnett square practice. For the duck activity, I have given you 5 traits, and I will send the parent traits as necessary, but your goal is to draw the duckling based off of what 5 traits you get. So you can use a simple number randomizer for this, set to from 1 to 4, 1 to 4 being the different Punnett square squares you're going to use. So for example, if we had a trait that was, let's say dominant versus recessive, let's say that this codes for beak color, we will have big B, big B. So we have a 50% chance that we get a orange beak and 50% chance we get a red beak. If we roll, I'm going to use dice because that's what we're using in class, we get box 1, which means that our duckling, our offspring, will have big B, which would translate into an orange beak. So each trait has a different dominance associated with it, whether it's sex-linked, whether it's incomplete, complete, or codominant. So you'll want to figure that out. So that is where I will end it today. The video for pedigrees will also be coming out here shortly. So look forward to that, because that will take everything that we just learned about Punnett squares and kind of apply it to how we look into families and their genetics.